Hello and a very good day to everyone. I am Nur Fasiah binti Muhammad Fauzi from Bachelor of Science Marine Biology. Today, I would like to present to you about metal contamination using polymer soda dispenser, which is known as March clamp as bioindicator. Luckily, polymer soda dispenser, also known as marsh clamp. This marsh clamp, formerly known as Geloina dispenser, is found buried in the stiff mud of the landward fringe of mangrove. It is well adapted to this habitat and has the ability to resume filter feeding rapidly when inundated. The research background of these studies are anthropogenic activities such as industrialization, agriculture practice, and the discharge of domestic sewage profoundly influence the Kelantan River. Polymesoda expensa live in association with mangrove forests and can tolerate long periods of the low tide. The marsh clams feed the filtering most of the suspended particles found in its environment. Due to their feeding habit, they are exposed to a variety of pollutants such as trace metal. The consumption of marsh clams contaminated with heavy metal may be detrimental to human health. Justification: the marsh clams have the ability to accumulate metals in its tissue, making it an useful indicator for monitoring heavy metal levels in the marine environment. Second, there is a high probability that these lethal wastes accumulate in the sediment and marine organisms, including shellfish, which can then cause human health problems. For example, large doses of consuming zinc taken by mouth even for a short time can cause stomach cramps and vomiting. Third, this study will add in knowledge on the occurrence of trace metal contamination will affect the polymer soda distribution. There are three objectives of this project, which are: first, to compare the distribution and abundance of polymer soda dispenser at industrial area and non-industrial area. Second, to analyze the effectiveness of marsh clamp in monitoring the presence of heavy metal in the estuary. And the third is to investigate the human health risk of heavy metal contamination in marsh clamp. This slide shows the sampling site for this project. This project will be carried out at Kelantan and there are four stations along the Pangatu River. Station 3 and 4 located near the industrial area of Pangkalan Chepa, while Station 1 and 2 located at non-industrial area. These clamps are used as bioindicator in monitoring the presence of trace metal at the target location. So there are three main methods of this project, which are sample collection, sample analysis, and analytical method. For sample collection, first, the live specimen of marsh plant will be collected in the mangrove area along the Pangatu River, Kelantan during July 2022 in four different sampling stations. Second, sampling station 1 and 2 will be located before industrial area. Meanwhile, station 3 and 4 will be located near to the industrial area of Pangkalan Chepa. Third, sediment attached to the marsh clamp will be washed thoroughly by the in situ water. And fourth, all samples must be stored in the low temperature before the sample will be dissected in the laboratory. For sample analysis, the sample will be washed with the distilled water in the laboratory, and then the sample will be classified. Weight and the length is measured using vernier caliper. After, all the soft tissue will be removed from their shell by using ceramic scalpel blade, and then thoroughly is rinsed to remove any extraneous impurities. And lastly, the total weight and wet weight will be recorded on the data sheet. For the last process, which is analytical method, the soft tissue will be weighed approximately to 0.05 gram in Teflon digestion container. Suprapyl nitric acid will be added into each of the sample and let it to pre-digest. After that, standard reagent and the blend will be done simultaneously. The container will be covered and placed in the stainless steel bomb, which will be sealed with screw closure to avoid any acid leakage and stored in the oven. Next, the sample will be heated in oven temperature for 7 hours at 100 degrees Celsius in order to digest the sample completely. After cooling process, the solution will be transferred into a centrifuge tube diluted with deionized water to 10 milliliter. The metal content then will be measured by inductively coupled plasma mass spectrometry. Then the statistical analysis, the test of SPSS software will be used to investigate the variation in the trace element, concentration in the organism, and the sampling site. Lastly, the level of significance of the statistical analysis will be set at 0.05. As you can see, this is a Gantt chart of this project that will be carried out. The expected result from this project. First, the expected result should be the concentration of the heavy metal at fall station. The level of trace metal in industrial area station will be give a higher concentration compared to the non-industrial area station. The previous research has proved that human activity at a specific point of time may contribute to the accumulation of trace metal in marine organisms. Next, the MASH claim will be used to observe the heavy metal pollution at the market station as they are an effective bioindicator. Yusuf has stated that in the past, MASH claims have been used as biomonitor to evaluate the marine habitat ecological condition. So the consumption of clams that have been bioaccumulated with metallic non-essential can cause harm even to the low concentration of organism and may endanger human health. So there are references that I have been used to complete this project. That's all from me. Thank you.